Good morning, everyone. Hope you all are doing well. So we were talking about transcription. On Monday, we talked about transcription in prokaryotes, and we covered initiation and elongation. Today, we will talk about termination of transcription in prokaryotes, and then we will move on to transcription in eukaryotes. We are following chapter 13, pages 448 to 457. There's a lot of details in the book. You don't need to know every single detail, but you need to know in general transcription factors and how these are they recruit RNA polymerases to the genes in eukaryotes. And most of the part that we will cover in the lecture is what you need to know. Okay. All right. So before moving on to termination of eukaryotic transcription, I would like to review the transcription a little bit, so we all are on the same page, just to make sure. So here, this is the pole, the E. coli RNA polymerase. Remember, prokaryotes have only one RNA pole, so this RNA pole will be synthesizing all different kind of RNAs in prokaryotes. So this is an example of E. coli polymerase. It has this is holoenzymes. It has all different components: alpha, beta prime, omega, and sigma. Transcription in E. coli we can divide it into 3 stages, just like DNA replication. It's initiation, elongation, and termination. We talked about initiation and elongation in the lecture on Monday. So here, the sigma binds to the promoter complex, -10 and -35 sites, so Ribnau sequence and -35. This is the transcription start site, +1. The sigma, 2 subunits of sigma 2, and sigma 4 bind to these 2 different sequences on DNA. Once its sigma binds to this DNA, it will open, so this whole holoenzyme is recruited here, and it will open the DNA. This is now your open promoter instead of the closed promoter. That will be before opening. Once about 9 to 10 nucleotides are added to the from transcription start site onwards, all that happens when sigma is still attached to the holoenzyme, and the holoenzyme is still at the promoter site. Once we have about 10 nucleotides added, then sigma will be released, and at this point promoter, this RNA polymerase, will start moving along the DNA sequence, opening a short sequence at a time, and making this, transcribing the mRNA sequence, transcribing the RNA sequence. This is the elongation, and at some point it will reach to the termination sequence. The termination sequence that's where transcription will be terminated. We also talked about proofreading. There are 2 different mechanisms of proofreading in transcription in prokaryotes. Pyrophosphorylatic editing, where you use the recently released pyrophosphate in order to replace a wrong nucleotide, and hydrolytic editing, which act as more as a 3 to 5 prime exonuclease activity, which is very similar to what we talked about DNA pole, but this exonuclease activity is stimulated by GRE factors. These GRE factors, they can both enhance the hydrolytic editing, and they also serve as elongation stimulating factors. So these GRE factors are already with this polymerase complex because these are elongation stimulating factors. There's another group of proteins that I mentioned briefly in Monday's lecture. These are called NUS proteins, or N-utilization substance. That's what NUS stands for. These NUS proteins join polymerase in elongation phase, and they also act as promotion and elongation factors as well as termination factors. So here you can see this is the NUS protein bound to this promoter, this polymerase here. This is the RNA polymerase polymerizing or transcribing this RNA. And one thing to keep in mind here is you see that this is all complexed with ribosome because this is prokaryotes, and in prokaryotes transcription and translation occur at the same time. As the transcription is occurring and the in the DNA. At the same time, that already transcribed part of the transcript will be translated in the ribosome. This is not the case in eukaryotes, because in eukaryotes RNA goes through post-transcriptional modifications, and we will talk about those today. We also talked about these TRCFs. If RNA pole stalls, then these helicases, these TRCF helicases, they can push the RNA through that problem, or it can stall it there or dissociate it there. What that means is RNA pole will be removed, so it's not making that transcript anymore. This TRCF is an helicase, but it can also perform the activity of polymerase removal and also recruits repair enzyme. So remember when we were talking about DNA repair, that's when RNA pole is making a primer. If it finds something, some issues, at that point it will recruit the repair enzymes. So this is what TRCF's function is. Okay, now moving on to today's topic. This is termination of transcription in prokaryotes. Termination it depends on the terminator sequence, what kind of gene we are talking about. Some genes have different type of sequence for termination. Some have a different another type of sequence. These are called either row independent or row dependent. So there are two different kind of sequences depending on what the gene has. Both of these mechanisms, they require hairpin formation. So here, this is a hairpin formation. Okay. So let's talk about row independent terminators first. These are also called as intrinsic terminator. So in these terminator, at the 3' end of the gene, that's towards the end of the gene to where it will be terminated, there is this inverted repeat. This is 7 base pair long inverted repeat. So this is the repeat here, 7 base pair, 7 base pair here. These inverted repeats are followed by 8 DTs. So here, these DTs or DAs, as you can see. These DAs, because this adenine will be uracil in the RNA, so this will make these uracil. So this has this dyad symmetry. Means they have these 2 inverted repeats, fused sequences in between, and then we have this multi-U sequence. So what happens is once when the template strand has these multiple A's, it will make multiple U's on the transcript, right? And before getting to these multiple A's or multiple U's, the sequence has to, the transcript has to copy these inverted repeats first. These inverted repeats form a hairpin loop as it starts moving into this transcribing these multi-T's. Because this DA, A on the DNA and U on the RNA, these are very unstable, these heteroduplexes, so the transcript dissociates from the template along with the polymerase, and that's the termination. So because once the polymerase is dissociated, transcription is complete, it's halted there, stopped there. Okay. So the row independent terminators, these are intrinsic terminators. They do not need any other protein. All they need is this inverted repeat sequence followed by multiple A's on the transcript, on the template. Okay. Row dependent require this protein called row. This row here, this is row protein. Row protein it's a ring-shaped protein. It has 6 identical subunits, so it's an hexamer. This row dependent termination is somewhat similar to row independent, where it also has these inverted repeats, but it does not have the 3' and but it does not have the DTs. So the 3' end does have inverted repeats that can make a hairpin, but they do not have this run of DTs. That's where the row protein comes, and row protein helps dissociate the transcript from DNA. So What happened in here in the row dependent termination as a row factor, or this row protein, which is an hexamer, it has an RNA helicase and ATPase activity. So it binds to row utilization site or RUT sites. These binding of this row protein to RUT sites is kind of similar to the actual, which is the RUT sites are close to where the termination point is, but not exactly at the termination point. It's the 5' of the termination point. Once the core polymerase stalls, because it reaches towards the end of transcripts, it has those inverted repeats. Once it reaches to those inverted repeats, the core polymerase stalls. Then row reaches there, and it allows the dissociation. Hopefully, what people think is very likely that it opens the fingers of the polymerase. Remember, those beta and beta prime make those fingers, make those pincers. So it probably opens those pincers. Okay, so this is row independent. Here it shows these NAS proteins. They bind, these are termination proteins. These will bind to this hairpin, that makes this hairpin, and this multiple AU, this heteroduplex will make it unstable. And then it comes off or dissociate from the DNA, this whole RNA polymerase. So RNA transcript will be stop transcription here as well. In row dependent, row protein is required. In this example, it doesn't show hairpin, but row dependent can have a hairpin. In most cases, it does have a hairpin. This is the RUT site, row utilization termination site, so that's where the row protein will come and bind. This row protein then moves to RNA polymerase where it is stalled, where it stops because of those, because it reaches to the end of the DNA unit where there's a stop codon, but it also because there is usually a hairpin. This row protein, once bind to this RNA polymerase, will help dissociate the RNA polymerase, likely by opening the fingers of the polymerase. Okay, so that's the termination of transcription in prokaryotes. Now we're going to move into transcription in eukaryotes, and transcription in eukaryotes is a bit different from prokaryotes, but the RNA polymerases are very similar in both prokaryotes and eukaryotes. One of the major differences in prokaryotes and eukaryotes transcription is that prokaryotes have only one RNA polymerase, whereas eukaryotes have multiple different RNA polymerases. Pol 1, Pol 2, Pol 3 are they polymerize different classes of RNA. We will talk about these. We will be focusing most on RNA Pol 2 because this is the one that polymerizes mRNA, messenger RNA. Plants have two additional polymerases, Pol 4 and Pol 5, that we will talk about. So here you can see that the prokaryotic bacterial core, prokaryotic, or in this case they divide into bacteria and archaea. Here we've only talked about bacteria, so I will just mention this. Beta, beta prime, alpha, and omega, these are the core polymerase, part of the core polymerase, and similar to these in eukaryotes are RPA1, RPA2, RP C5, C9, B6, and here Pol 2 that we will be talking the most about is RPB1, RPB2, RPB3, RPB11, RPB6. RPB stands for ribosomal protein B, which means B means 2. That's what, because it belongs to Pol 2. That's B. Usually what belongs to Pol A, Pol 1 is A, Pol 3 is C. Okay. So eukaryotic transcription, there are, as I said, unlike prokaryotes, eukaryotes use different RNA polymerases to synthesize different classes of RNAs. Ribosomal RNA is transcribed by RNA Pol 1 in nucleolus. mRNA is transcribed by RNA Pol 2 and as heterogeneous nuclear RNA, and it's unspliced mRNA precursor. We will talk about that. The RNA pol 2 also transcribes the small nuclear RNAs. Pol 2 requires ATP. tRNA or transfer RNAs are transcribed by pol 3. Also the 5S subunit of ribosomal RNA. Usually all small RNAs are transcribed by tRNA. Transcribed by pol 3, I'm sorry. Plants have additional RNA polymerases, pol 4 and pol 5, to transcribe the small interference RNAs. And here again, so these are examples of 3 of these. So pol 1 usually transcribes ribosomal RNA. Pol 2 transcribes mRNA and small nuclear RNA. Pol 3 usually transcribes 5S ribosomal RNA and all transfer RNAs. Okay, so let's look at RNA pol structure. RNA pol structure, they are complex. There's a big complex of 12 to 14 subunits, as you saw here. These are a couple of subunits are given names, and there are additional subunits as well. So altogether, these make about 12 to 14 subunit. These RNA pol 2 subunits are usually labeled as RPB for RNA polymerase B. RNA pol 1 and pol 3 are made of homologous or orthologous subunits, but they encode their own genes, so they are usually called A and C respectively. The core enzyme, or RPB, core enzyme is analogous to, in coenzyme RPB1 is analogous to prokaryotes RNA pol beta subunits, which binds to DNA. The CTD, the carboxy terminal domain phosphorylation, is required for the transition to elongation in eukaryotes. This is not the case in prokaryotes, so that's another difference. To move from initiation to elongation, the CTD phosphorylation is needed. CTD is a highly repeated serine-rich 7-mer. So here, this is the CTD of RPB, of the coenzyme RPB1. This RPB1, this phosphorylation, here you can see that this is a 7-mer, so seven amino acids are there. So these seven amino acids are repeated 52 times in humans. Phosphorylation of the CTD tail is required for promoter escape and elongation. So here, this is phosphorylation and dephosphorylation of serine at the 5th position is very important. This phosphorylation status of RNA pol will guide whether it will go into elongation or not. RPB2 is analogous to prokaryote RNA pol beta subunit, which is the major function of this RPB2 is polymerization, and RPB3 is similar to alpha subunit. It has 2 molecules per core. So we talked about promoter structures, promoter structure in prokaryotes, where the promoter is as Pribnow box, or -10 and -35. -10 is required for transcription. -35 is not required, but it's usually present in promoter. That's the core promoter we talked about. But when we talk about this promoter structure, the classical structure of promoter in eukaryotes, it's going to be a little more detailed, a little more complex than the prokaryotic core promoter. One of the important sequence in eukaryotic promoter is that you might remember is Tata box. The Tata box is at -25, which is this Tata box is similar in sequence and function to that of Pribnow box in the prokaryotes. However, Tata box is not found in all genes. It is some of the housekeeping genes and some of the developmental genes usually lack Tata box. Instead of Tata box, they use a different sequence as their core promoter. Theta box is usually found in tissue-specific genes. Another important sequence for this in promoter structure is BRE, or TF2B response element. TF2 is a transcription factor 2. This TF2B response element is at around -32 to -37 position, just upstream of theta box. This binds transcription factor 2B, which is a general transcription factor. Initiator sequence, or INR, it starts at -2. It has a consensus sequence of YYANWYY, where Y is any pyrimidine or W is either A or T, and N can be any 4 of the nucleotides. Initiator binds to transcription factor 2D. So generally it has a weak function just by itself, but it can stimulate theta box or a GC box. A GC box is far upstream. It can be several hundred nucleotide away from the core promoter or the transcription initiation site. DPE is downstream promoter element. This is downstream of transcript start site of +1. It stimulates transcription of genes such as housekeeping genes. So one thing to keep in mind, the genes that have DPE usually lack theta box or other way around. So the genes will either have theta box or the DPE downstream promoter element. All these other sequences are pretty conserved. So this is the BRE or TF2B response element here. This binds to TF2B transcription factors. Theta binds to theta binding proteins, TF2D along with the TF2D transcription factor. TF2D transcription factors bind to a number of different sequences, and we will talk about this just in a minute. And this is the initiator sequence. That's where the initiation will start. So this initiation will start at +1. So that's right here. TF2D, this can be separated into 9 subunits. These are TBP, the theta binding protein, and 8 TAFs, or theta binding protein associated factors, or TAFs. These TAFs differentiate. This is called TAF, usually TAF2, because just to differentiate it from TAFs of pole 1 and pole 3. TBP is the name stands for theta binding protein, but it is doesn't only bind to the theta boxes, but it also binds to theta-less promoters. For example, class 1 and class 3 promoters that do not have theta box element. This TBP binds to those sequences as well. This TBP contains a C-terminal basic DNA binding domain, which is important. Because why it's important is because the sequence will bind to the DNA first, and the binding of TBP usually is enough to change the structure of DNA, or change or bend the DNA that it will allow other proteins to come and bind, and it will open the DNA sequence. The TBP footprint is about 20 nucleotides over theta box, so it binds to theta box, including additional nucleotides, a total of 20 nucleotides. It binds to minor groove of DNA, and it makes the DNA bend at about 80 degrees, causes the local helix melting. Because the theta box is rich in AT base pair, and AT base pair melts relatively easily because they have double bond instead of triple bond of GC. That's why it is assumed that the theta box is conserved in many genes and many organisms, many species, because of this ease of melting of DNA molecule at the AT rich regions. So here again, this is your TF2D. TF2D also include TBP. These TBP and TAFs together make TF2D, and these bind to multiple different sequences. TAFs, these theta binding protein associated factors, these modulate the ability of TBP to interact with the promoter, especially the promoters that do not have a theta box. So these TAFs can recognize portion of the core promoters, and these will bind there. So for example, in this here, this TBP binds to data box, but these TF2D, these contains both TBP as well as TAFs. These can bind to other sequences as well. TAFs helps them bind these TBPs to different sequences that are not data box. Some of the promoters have GC box. In addition, it can be in addition to data box, or that's just the GC box. GC box also interact with TBP through another TAF. This is called TAF2 remember, TAF2 is because these are TAFs associated with the pole 2 1110, and this TAF2 1110 also binds to SP1. SP1 binds to GC box, so SP1 will, this is a specific transcription factor, it's not the general transcription factor. So this SP1 will bind to GC box, and then this GC box will interact with TBP through this TAF2 110, which binds to SP1. So these TAFs can be sometimes co-activators, because these are not just activating the data box, the core promoter, but these can also activate the upstream promoters, for example, the GC box. These TAFs, so these TAFs, these also allow interaction with specific transcription factors. So say, for example, this SP1 we just talked about. SP1 is a specific transcription factor. It's not a general transcription factor. General transcription factors are these transcription factors here, TF2B, TF2Ds, which are required for the core promoter activation. Specific transcription factors vary from gene to gene because these are for gene-specific transcription factors, whether this gene has to be overexpressed, underexpressed, that will determine by these specific transcription factors. The promoters with data box, the TF2D act as an assembly factor, and promoters that use these additional sequences, for example, GC boxes, this SP1. SP1 is one of the examples that can act as assembly factor and recruit TF2D. Okay. It's class 2 promoters where RNA pole 2 binds. So RNA pole 2 binds to class 2 promoters. These class 2 promoters, they usually have, they either have a data box or a DPE. So the ones that will have data box will not have DPE. The ones that have DPE will not have data box. So most of the housekeeping genes, for example, have DPE instead of data box. Most of the tissue-specific genes have data boxes instead of DPE. Sometimes genes have upstream element. The most common upstream element is GC box at about -100. So this GC box will be, so if we look at here, so if this is 1, data box is at about -25. At about this GC box will be up further upstream at about -100. They can also have a CCAAT box. The CCAAT box is also, in this example here, it shows a GC box as well as CCAAT box in this promoter. These CCAAT box is usually around -80, -85 base pair upstream of the transcription start site. GC boxes are bound by these SP1 transcription factor that we just talked about. These are specific transcription factors. These GC boxes are usually found in data box less promoters, for example, housekeeping genes or developmental genes. So if you don't have data box, you might have a GC box in addition to the downstream promoter element. CCAAT box, these are bound by CTF or CCAAT binding transcription factors. These are either the CTF or sometimes these are bound by CEBP or CCAAT enhancer binding protein. So when there are these CCAAT box binding to enhancer binding protein, that means the expression of that gene is enhanced because they are using an enhancer in addition to their promoter. It's really important to understand, to know your sequences, promoter sequences in your gene, in order to know whether this gene is going to be highly transcribed or not as much transcribed. These are also very important. Promoter sequences are really important to be understood, especially when maybe it's a tissue-specific gene and you want a promoter for that gene to be able to express your gene of interest, maybe in a very tissue-specific manner. Say, for example, we have this gene. Say, for example, vitellogenin is a yolk protein. This yolk protein is the gene only expressed in fat body or the liver or adipose tissue. That's where the fat will be synthesized. So if you want to express your gene in a way that it only expresses in the fat body or in the liver, you can use the promoter of this gene specific, the specific gene that only expresses in that tissue, and that way your gene, under the influence of that specific tissue-specific promoter, will also only express in that tissue. So when people make transgenic animals, when they don't want the gene to be globally expressed, they want it to be very tissue-specific expression. In that case, you need these promoter sequence of a tissue-specific gene as well. So that's why it's very important to understand the promoter structure and what kind of promoter is there. Okay, now what we talked about so far is we talked about core promoter. So core promoter is the minimal portion of the promoter that is required to properly initiate transcription. So this is your gene will not express without the core promoter. This is the minimal promoter that is needed. This minimal promoter has a bunch of different criteria that need to be followed. And your core promoter includes the transcription start site, or TSS. That is required because that's where transcription will start. It will also have elements that are directly upstream of the transcription start site. These elements that are directly upstream transcription start sites are a binding site for RNA polymerase. It need to have general transcription factor binding sites, for example, either a Tata box, a BRE. These need to be there. They need to have some other elements or motifs need to be present in order for that promoter to be active. For example, the TF2D sequence binding sites. And just to keep in mind, there is no universal element found in every core promoters. These core promoters differ in different genes. For example, a good example here will be presence of Tata box or a downstream promoter element. Some genes have Tata box, some genes don't. Some genes have GC box, some genes don't. So these core promoter structure vary from gene to gene. There is no universal core promoter. There are some elements that are conserved. For example, a TSS and a RNA pole binding site will be conserved in all promoters, but what other general transcription factors are, these sites might vary. Proximal promoter, these are not present in all genes. The proximal promoter sequence are upstream of the gene, approximately 250 base pair upstream of the start site. These bind to specific transcription factors. So these proximal promoters are usually present in, for example, housekeeping genes or the genes that are highly expressed, but not in just the normal expression of genes. Distal promoters, these distal promoter sequences are also upstream of the gene, and these contain additional regulatory elements. Usually they don't have as much of influence on transcription as the proximal promoters do. These distal promoters are anything that are further upstream, but these are not enhancers. They also bind to specific transcription factors. So the proximal promoter and distal promoter is not the required promoter sequences, but the core promoter is required, is the minimal promoter. Okay. Proximal distal promoter help with the higher expression of the gene, but the core promoter needs to be there. Okay, now let's talk about the mechanism of transcription in eukaryotes. So far we talked about what the RNA pole looks like in eukaryotes, what the promoter sequence look like in eukaryotes. Now the pole has to bind to this promoter sequence in order for transcription to begin. So the first is pre-initiation complex. Pre-initiation complex is a complex of 6 proteins. These 6 proteins are transcription factor 2A, B, D, E, F, and H, and RNA pole 2. So you can see here that in eukaryotes there are many transcription factors, general transcription factors that are required for initiation of transcription. So here, let's look one by one. TF2D. TF2D binds at the data box via the minor groove. Remember, it kinks the DNA to about 80-degree angle where it binds, and that angle will lead to melting there. TF2A and TF2B bind after TF2D binds. So first the TF2 will come and bind to data box, then TF2A and TF2B will bind. TF2A binds to the N-terminal portion of TF2D, whereas TF2B binds more towards C-terminal of the TF2D, and TF2A stabilizes the transcription factors to the DNA molecule. That's kind of similar in function to the sigma factor, where sigma binds to minus sigma 2 and sigma 4 binds to minus 10 and minus 35 and keep the whole RNA pole there. TF2B acts as an adapter between TF2D and RNA pole. It will bind towards the C-terminal portion of the TF2D, and it positions itself in a way that it will place RNA pole 2 downstream of data box. So let me bring this here. So this is your DNA. This is the promoter sequence. This is data box here. So the initiator will be somewhere here, and the other sequences will be upstream. For example, BRE will be upstream. TF2D along with the TBP, the data box binding protein, will come and bind here at data box. Once this binds, then TF2A and TF2B will come and bind. TF2A binds more towards the 5' end, and it will stabilizes this sequence here. TF2B binds more towards all the way towards 3' of this TF2D, and this binding of this TF2B will bring the pole and put it in a position that it's at 3' of the data box. So data box is here, pole is right there. So this pole binds to this sequence. Pole has this tail. This tail is very important, and we will talk about this tail. The RNA pole doesn't really just come and binds. Once this TF2A and TF2B they position themselves onto the DNA molecules on the TF2D, RNA pole is possibly already bound to TF2F or transcription factor F will come and bind to this where everything else is. It's TF2B will bind to this pole here. Okay. This then TF2H and TF2E will come and bind behind the RNA pole too. So these are bound to RNA pole here. TF2H is very important in eukaryotic transcription because TF2H it has many subunits, and these subunits function as a protein kinase. What does that mean is this TF2H is the one that phosphorylates CTD of RNA pole 2, this tail of DNA pole 2. And remember, this phosphorylation will move the complex from initiation into elongation. Remember, this is rich in serine. Those 7 mers that have been repeated multiple times, these 7 mers, these will get phosphorylated here. Some of these, for example, the S5 that we talked about, will get phosphorylated, and then this complex will move, or the RNA pole will move from here onto the DNA molecule. TF2H stays attached to the RNA pole. Okay, so this TF2H is downstream of RNA pole 2. It uses energy of ATP hydrolysis. It stuffs the DNA into polymerase active site. That's at the initiation. During this initiation, the stuffing of DNA, similar to prokaryotic DNA that we talked about, when the pole is still at the promoter, the small portion of DNA up to 10 nucleotides will be brought in the RNA polymerase, and the nucleotides will be added there before the pole moves from the promoter. Before it detaches itself. So similarly in eukaryotes as well, this TF2H will bring the DNA into the polymerase active site. It will this will cause the strand separation of the DNA molecule. The double-stranded helix will open, and then the polymerase RNA polymerase will put the RNTPs there according to the sequence. This will result in an open promoter complex. Polymerase is still bound to the promoter at this stage. Okay, so here this is TF2H. TF2H brings this DNA molecules here. It makes this open complex. This will polymerize here, and then this when it's still bound to DNA. Once some of the nucleotides are formed, this will remove from this complex here and will move on to the DNA template to make RNA. RNA is, again, just like prokaryotes, RNA molecule does not stay hybridized with DNA, although it will rather come out of the RNA exit channel of the pole. This is that phosphorylated tail of RNA polymerase, and we will talk about how this is quite important, not only in initiation and elongation, but also in termination. As you remember that RNA in eukaryote has to go through post-transcriptional modification, and one of the post-transcriptional modification is adding the 5' cap. This 5' cap will be added as soon as RNA comes out of the polymerase, and we will talk about that in a minute. So here this shows that how this is the core promoter, right? So when it's bound to the core promoter, this is what it looks like. But that's not the only thing. It can bind to the activators. It can bind to the proximal promoter or distal promoter or GC box that is further downstream. So the further downstream sequences are brought to the same place by these mediator complex. So mediator complex will bind to these is really a part of one of the transcription factors, and this mediator complex has these proteins that will bind to the activator sequences of wherever those sequences are. These also bind to chromatin remodeler through these HAT histone acetyltransferases, and these can change the chromatin structure as this transcriptional bubble move along. Okay. In initiation, so we talked about pre-initiation complex. In initiation, RNA pole 2, we are using RNA pole 2 as a model. Here we're not talking about RNA pole 1 and 3. The major differences between eukaryotes and prokaryotes initiation include in eukaryotes, RNA pole doesn't scan the DNA and posit promoters, which it does in prokaryotes. Remember, the RNA pole will scan and find the correct position because those sigma subunits, those can find the correct position, correct sequence. If they bind to a wrong sequence, it will be dissociated, but if it finds the correct sequences, it will stay there. In eukaryotes, that doesn't happen, as the transcription factors are first assembled onto the DNA sequence, and these transcription factors bring the RNA pole to transcription start site. These other proteins, these transcription factors, these assemble at the promoter first, and these recruit RNA pole in eukaryotes. DNA helix melting requires ATP hydrolysis. but RNA pole doesn't open the double-stranded DNA, and this DNA helix melting, this ATP hydrolysis is also carried out by these transcription factors, because it's not the RNA pole that opens the double-stranded DNA. Transition from initiation to elongation in eukaryotes require phosphorylation of subunit of RNA pole to core, and that's the tail, that subunit. In eukaryotic RNA, polymerases require transcription factors to assist them in binding, and these perform a very similar role to the sigma factor of prokaryotic polymerase. But in eukaryotes, instead of one of the subunit of polymerase, instead of that, there are transcription factors that are helping with the same function. General transcription factors bind promoters. These are required to form this pre-initiation complex. These can recruit RNA pole 2, and they can stimulate transcription, although the transcription will be usually pretty weak if the promoter only has general transcription factors. So that means if the gene expression is low, those genes will have only general transcription factors, because for low transcription expression, it doesn't require any additional sequences, any activators or other regulators or distal or proximal promoters either. Specific transcription factors, these are usually the activators, and these are the proteins that bind to response elements. This can be enhancers or activator binding sites, and these stimulate transcription by assisting the assembly of general transcription factors to the specific transcription factors. So here, this is the core promoter, general transcription factors. These, when these are bound to activators, these will be via the specific transcription factors. So the genes that has these specific transcription factors, these bind to these activators, they will have higher expression. Now, elongation of transcription. RNA pole 2 needs to be, the tail needs to be phosphorylated by transcription factor 2H, because TF2H is a kinase. It adds the phosphate group on the amino acids. Once this phosphorylation happens, then the RNA pole will dissociate from the initiation complex, and it will move on to the DNA. This TF2H remains with RNA pole 2 during elongation, as I mentioned earlier. The polymerase binds to elongation factors, not the TF2H because that's already bound, but there are other elongation factors similar to what we talked about in prokaryotes, has this GRE factors. These are elongation factors. In eukaryotes, there are different elongation factors. The function of elongation factors is to increase processivity of the RNA pole. That means the RNA pole will have higher processivity. It will be able to catalyze multiple reactions in one catalytic event if there are elongation factors. It also increases fidelity. That means it checks for the correct sequence, correct nucleotide added to the growing chain, growing transcript. So it checks the fidelity, and it can also modify the transcript by capping or splicing or adding poly A, etc. TF2S, this transcription factor 2S, here, this associates with RNA pole 2, so this is one of the elongation factors, associates during elongation, and it facilitates 3 to 5' exonuclease or proofreading activity. So it's similar to these GRE factors of prokaryotes. It uses the hydrolytic editing. That means it can remove the wrong nucleotide in order and add a correct nucleotide through this 3 to 5' exonuclease activity. Fact, this is facilitates chromatin transcription. These proteins, these are the dimer of a protein, and these proteins can remove H2A, H2B dimers from nucleosome upstream of RNA pole 2. So if the transcription is moving and it comes across a nucleosome, these FACT proteins can remove the remove the histones so that the RNA pole can move through nucleosomes. So once it's removed the histones, then the DNA opens there, because remember the DNA in the nucleosome that's coiled on the histones. Once you remove the histone from there, it's already negatively supercoiled, so it's already loosely bound. Okay, now RNA processing. Once you have, so now we talked about RNA pre-initiation complex, initiation and elongation. Now as soon as I mentioned earlier, as soon as the RNA 5' end of RNA comes out of RNA pole, the cap needs to be added there. So that's the first event of RNA processing. In eukaryotic RNA, the eukaryotic RNA need to be processed before it leaves the nucleus, and the processing are 5' cap, addition of 5' cap, polyadenylation, and splicing. Splicing, if you recall, the DNA, the gene, has both exons and introns, but the mRNA will only have exons, so the introns need to be spliced out, so that's splicing. Polyadenylation is addition of adenine or A's at the tail end or 3' end of mRNA, and that's the specific feature of mRNAs, that they have these poly A tails. So capping involves addition of a modified guanine base to the 5' end of RNA. So if you remember DNA replication or transcription of RNA, they both move in 5 to 3' direction. So it's always goes add new nucleotide on the 3' end, but when we are adding a cap, that cap needs to go in the 5' end of the transcript, not on the 3'. It adds this 5'. This adds this modified guanine. It's a methylated guanine. And it also it's in a very unusual way. It's a 5' linkage that involves 3 phosphate groups instead of 1 phosphate group. Here. So RNA, as I mentioned, RNA is capped as soon as it emerges from the RNA exit channel. So here, so this is your transcript. So transcript is moving in this direction, 5 to 3' direction. This is the guanine that will be added to the 5' end. So this is 5' and this is the alpha, beta, and gamma phosphates. This alpha phosphate here, this RNA triphosphatase enzyme will be utilized here to add this cap there. So this cap, this will remove 1 phosphate from here, from the already existing these 3 phosphate group, will remove the gamma, and it will attach itself to the alpha of this incoming guanine, resulting in 3 phosphates here, 5' linkage. This is what the 5' linkage is. So RNA triphosphatase enzyme will catalyze this reaction, and then guanylyl transferase will transfer this guanine here, will make this 5' linkage, and this, once this 5' linkage is made, once this guanine is added to the 5' of this transcript usually starts with A, because the first amino acid is in most protein is a methionine, so it's AUG. So A is the first nucleotide in a transcript. And here, then this methyltransferase will add a methyl group on the 7th position on this guanine. So this is now a methylated guanine, which is attached to this transcript by an unusual 5' to 5' linkage. This is your 5' cap. This 5' cap is added as soon as the RNA emerges from RNA exit channel of the polymerase. So there are 3 enzymatic steps here. Phosphate group is removed from the 5' end of the transcript by RNA triphosphatase. The GMP moiety is attached here by this guanylyl transferase, and then the nucleotide is modified by methyltransferase by the addition of this methyl group. Then here, so this is the pre-initiation. There is no phosphorylation of CTT tail. At the capping event, there is phosphorylation of serine 5. This leads to promoter escape as well as capping. So promoter escape means now the RNA pole has left the promoter and it's on the DNA molecule. It's moving. It's in the elongation phase. So this is this promoter escape and capping will occur when there is a phosphorylation on the serine 5. This serine 5 phosphorylation, this serine 5 position is dephosphorylated during elongation. In elongation, there is no phosphorylation on S5. However, there is phosphorylation on S2. So this dephosphorylation of serine 5 in CTD dissociates the capping machinery, and the cap formation is now complete. Termination of transcription. So we will now talk about once the elongation is complete, when the sequence reaches to a stop codon, what happens at that point. So polyadenylation of the 3' end is associated with the termination of transcript in eukaryotes, in eukaryotic mRNA synthesis. What happens here is the CTD tail. Remember that CTD tail of RNA pole, that tail is involved in recruiting enzymes necessary for this poly-A synthesis. There are two protein complexes here. So this is that RNA pole tail. This is a phosphorylated tail, remember, because it phosphorylates depending on whether it's an initiation or elongation phase. Once it reaches the poly-A signal sequence in the DNA, once the pole reaches towards the end of the transcript, the CTD tail will have these enzymes. It will recruit these enzymes that are required for poly-A sequence, and these two of these enzymes are a CPSF protein complex. Two protein complex called CPSF is a cleavage and polyadenylation specific factor, and CSTF. So here's the CSTF, here's the CPSF. CSTF is cleavage stimulating factor. So the tail has these enzymes that will make these proteins, these sequences that will make these proteins. These proteins will then added to the poly-A signal sequence in RNA. Once these are attached to this growing poly-A tail, it will lead to cleavage, because that's what the protein complex does. This is cleavage stimulating factor. This is cleavage and polyadenylation specific factor. This leads to addition of these proteins to the growing mRNA molecule once it reaches to poly-A signal sequence in DNA. So it's copied this poly-A signal sequence in DNA, which is before the termination. These proteins will come and bind to this poly-A signal sequence in the mRNA as well. This will lead to mRNA cleavage. Once the mRNA cleavage happens, the CSTF, this cleavage stimulating factor, this dissociates, whereas CPSF stays associated with that sequence. It already have 5' cap here. The CPSF stays there. The CPSF then recruit another protein called poly-A polymerase. This is a specific polymerase or PAP that will add this multiple A's on the 3' end of the mRNA. So once these CPCF, CSTF are bound, other proteins are recruited, leading to RNA cleavage and then polyadenylation. This poly-A polymerase, which uses ATP, adds about 200 A's to the 3' end of RNA. So it starts adding. This is the specific. This is a specialized RNA polymerase called poly-A polymerase. This poly-A polymerase will add these A's. Poly-A binding proteins, these are similar to those single-stranded DNA binding proteins, will come and bind to these this poly-A tails, so that it will not move onto itself. There's additional poly-A binding proteins. Once this reaches to about 200 nucleotides, 200 adenines, these the PAP and the CPSF, these will dissociate from from the mRNA, and at this point the poly A tail has been formed. So now the RNA already have a 5' cap and poly A tail. Now the next event for the RNA will be to splice the introns. So at this point the termination has happened right here when these CSTF and CBSF bind to poly A signal sequence in RNA, and this CSTF leads to the cleavage of mRNA. And at this point the RNA pole will dissociate as well. Okay. RNA pole 1 and pole 3 recognize distinct promoters, but they still require these Tata box binding proteins. So that's why, as I said, it's a little misnomer, because these proteins don't always bind to Tata boxes. These can bind to other sequences as well. So because here you will see that the pole 1 and pole 2 don't have Tata boxes per se. Pole 1 transcribes ribosomal RNA. The promoter for ribosomal RNA gene consists of this upstream control element, or UCE, and the core element or core promoter. This core promoter is more or less similar to the core promoter of pole 2. Pole 3 promoters are very different. They transcribed transfer RNA and 5S RNA of ribosomal RNA. These are unusual promoters because the promoter is located downstream from the transcription start site. So this is the transcription start site. This is the example of a 5S RNA promoter. This is the transcription start site, and these are the different elements that will be needed for promoter. These are the promoter elements. Similarly, this is the promoter for tRNA. This is the transcript start site, and these are the promoter elements that will be used used for transcription initiation. And that's all we're going to talk about today. On Friday, I will talk about we will talk more about transcription, especially RNA processing, as that will be the topic for Friday. Another thing I would like to mention that on Monday, instead of starting a new topic, I would like to have a review of all the material that we have covered so far. So if you have any questions, so go over all the materials over the weekend. If you have questions, send me the questions, and I will answer all of your questions on Monday before moving on to the next topic. So I want to make sure that we all are able to understand the materials well, so that you will not struggle during the next test. So please send me all of your questions starting from our lecture right after spring break up until Friday. Actually, I will cover Friday's lecture, and then I will answer the questions that you send me on Monday. Okay, and be safe.